So good morning, everyone. Congratulations for showing up this early. The first presenter is Kevin Slavin, who's currently the Chief Science and Technology Officer of the SHED, which is a multi-art center in New York opening in 2019. He's also the founder of Playful Systems at MIT Media Lab. And he also gave a TED talk titled How Algorithms Shape Our World, which you should watch, because today he will instead talk about urban metagenomics and how to map the invisible biological cloud of the city. And I think we can start with the question, what is urban metagenomics and what can we learn from it?
Great, thank you. Let's see what happens when, here we go. And good. Let's see what happens. I'd never heard that phrase before, congratulations for showing up, but congratulations for showing up. Actually, a couple things have changed here. I have to escape out. But I could tell you some jokes.
No.
But it's mirrored now, and but it can't be mirrored. I need separate display, like what I have. Or maybe you guys have jokes. That's good. I like that. Good. Okay. If you could see what I see. Okay, there we go. Okay, good. Okay. This is an old quote from a really fantastic writer, Philip Gorovich, who wrote, "This is what fascinates me most in existence, the peculiar necessity of imagining what is in fact real." And I'm going to use that as a starting point today to talk about the notion of urban metagenomics. And if you know what urban metagenomics are, then you should leave because you'll be bored. But nobody knows what it is because it's new. Okay, let me start with this. This is Bristol. This is an artwork by an artist named Maria Teresa Alves. And it's a work of art that she did for the city of Bristol that was what she called a ballast garden. And a ballast garden, well, to get into it, you'd have to know what the ballast is. And the ballast is it's the part of the ship at the bottom there where it needs to be weighted down. Otherwise, the ship can tip over. And if you're traveling across the ocean, as in the ages of colonization and exploration you were doing often, you needed to fill the ballast with whatever was local material just to keep the bottom of the ship heavy. And because of that, if you find shipwrecks in, for example, Florida, you'll find actually just the wood has rotted away. You'll just find big clumps of stones that had been at the bottom of the ship. So that's what a shipwreck looks like, a 16th century Spanish shipwreck in Florida. And I mention it because it's a good example of, these are basically Spanish rocks. They were brought over at the bottom of the ship from Spain, and they carried with them Spanish dirt that were sort of around those rocks. And in that Spanish dirt were, of course, Spanish seeds. And from those seeds, Florida grew. And from all the seeds, from all the ships that were crisscrossing in that period, and even today, what comes to life is what comes along. So when Maria Teresa Alves built the Ballast Garden in Bristol, she planted a garden made up entirely of the species of plants that are growing and live in Bristol as a sort of byproduct of the ages of colonization and exploration. So she planted marigold, which had come over from the Mediterranean, and Eruca sativa, and oats, and Ficus carica, and these are all quiet migrants. Nobody intended to bring those to England. Nobody planted them there. They actually sort of came over in the dirt that was in the ships. And I mention it because this action of hers, but the sort of larger action that lies underneath it, raises these kind of interesting questions about, from that period, who exactly colonized whom? Which colonization worked in which direction and to whose benefit? And it happened because no captain was ever really aware of how much of their cargo down there was alive. And I bring all this up today because, of course, as it was true for those captains, it's true for us. Neither are we. None of us are aware of how much life we carry within us. And I don't mean it in some sort of like hand-wavy, highfalutin sense. I mean that, I mean basically the gut and the 10,000 or so different species that live in each one of your guts. Slightly different between you, massively different if you come from far away, much closer to one another if you live together or if you have a dog, things like that. We'll get to that. But the point being that a massive amount of you isn't really you. And there are different ways to kind of figure out exactly how to estimate this, but depending on how you calculate it, it could be 50%, could be up to 90% of your genetic material isn't human at all. It's around 5 pounds of you. And the interplay between the you that is you, the human part of you, and those 5 pounds of all the other species that are inside you it's come to be understood as having at least as much to do with your life and your outcomes, your behavior, what your world feels like as any sort of genetic material that you happen to have got from your parents or whatever broad social experiences you have. The effects and interplay between what's happening here and the rest of us is still unknown. I would say that we have learned a lot as a species. We've learned a lot, probably in the last 10 years or so, but it's still kind of abstract. This is an article from not too long ago that a baffling brain defect is actually linked to gut bacteria, right? So think about that for a second, right? That there's the idea that there's something that lives in the gut that changes how the brain wires itself. And I could give you a lot of different examples of these. We don't really understand a lot of the how yet, and we're just beginning to understand the what, but we're learning. And we're learning in part because of the technologies around genomics, which is to say the technologies that allow us to understand the genetic material in us. So, if you want to find out what's in you from a genetic perspective, you could spit into a flask and you can send it off to a bunch of different companies and they can tell you what DNA is in there. And there's a couple different startups where you can send your poop and find out who's living inside you, and that there'll be much more of that in the future is quite obvious. And you can do that by sampling the spit or the poop, but the question is, what's this? And how do you sample that? And it's important to be able to understand what that is, because that gut biome that really determines who you are, that gut biome that is the essence of you, it's basically formed from 3 sources. It's basically an exchange with 3 sources in the world. It's formed in part by a bunch of bacteria that you get when you're born and move through the birth canal. It's in part through what you eat. But a lot of it, maybe most of it, is basically what you touch and breathe. And then the question is, what is that? And how do we find out about it? And all of this is possible as the sort of unexpected benefit of cheap genomic sequencing. I think that when most people think about the sort of lower and lower costs of cheap genetic sequencing, we think about it as like, oh, so it'll be much cheaper to find out whether I have, European heritage, or who knows what people are hoping to find in that. But that's a very human-centric view on this, right? The valuable part, in fact, of the collapsing price of sequencing genetic material is everything outside of humans. And that's what leads us to the notion of metagenomics. So to get your head around it, genomics, you would say, like, if I gave you an onion and said, what are the genomics of this onion, you can use genomics to do that. But if I gave you like a bowl of soup and you had no idea what's in there, you'd have to use a metagenomic approach, which means you'd have to extract the DNA from everything in the soup and then compare that to all the DNA of everything in the world, which is probably the largest data set, maybe outside of climate data, that you can imagine. It is basically, we are building and we have built the catalog of genetic material of everything on Earth And now the question is, where is all of that? Those things that we have cataloged, where are they, and how do we begin to understand where they've got to? And of course, statistically speaking, most of us as humans live in a very special kind of soup that we call cities, and we live in these sort of unaware of their live freight. We live in cities with a natural bias towards thinking of them as built for the interaction between humans and the built environment. That is, of course, not at all what's happening. And the question is how to even build an imagination of it. And so I was inspired in part by work from some good and old friends who did this years ago. They mapped the Wi-Fi signals in urban spaces using Wi-Fi receivers and lights and a camera, very clever technique from Timo Arnal. And it was a way to sort of imagine this invisible world around us. But the truth is that these invisible radio waves around us, like that we're soaking in right now, they're really nothing compared to the invisible living kingdom that's surrounding us even now. And basically over the course of a couple years when I was at MIT, started to try to figure out, well, how do we even understand what that is? And in the process of doing that, sort of discovered and made our own contributions to the notion of urban metagenomics, right? So if metagenomics is basically figuring out everything that's in the soup, urban metagenomics is figuring out everything that's in the soup called cities. So there are, that I'm aware of in the world right now, 4 map makers for this world. And I'll just go through these 4 different labs. The first one is a lab that I've worked with very closely in New York City, Chris Mason's lab at Weill Cornell, who started something called the PathoMap project. This was the first city-scale metagenomic profile. They had a couple $1 million, a couple hundred people, and they physically swabbed the surfaces of every subway station in New York City, which is a grim, difficult business. And they took everything that they swabbed, sequenced it, and then started to map, are there differences between the subway stations? Which actually gets at this larger question, which is sort of where we started, which is that in the same way that my gut biome is different than your gut biome, is Geneva's gut biome different than the gut biome of Brooklyn or Lagos or Tokyo? And so for Chris Mason's lab, they looked at it at a very fine resolution, which is it different on the east side of Manhattan than the west side of Manhattan? Is it different uptown than downtown? And the answer is that those subway stations are quite distinct, and that you start to see some very unusual and anomalous things once you can start to sequence the rest of the life that's in the subways. And so what they discovered, for example, is that even several years after Hurricane Sandy had flooded several subway stations, that those subway stations were, from a microbiological perspective, marine environments, even years later. That there had been this major shift in their ecosystem that is invisible to all of us, and yet affects us in all these ways that we're just beginning to understand. They also found a subway station, this is a story I don't really have time to tell, but they found a subway station that had a very specific bacteria that's only produced in certain kinds of fermentation, and were able to kind of backtrack several steps to realize that's where Korean Americans live, because Korean Americans eat a lot of kimchi, and the kimchi has live bacteria, and it moves through the skin, is left at the station, and then the rest of us get it. And that's a good thing, because it's actually very healthy, like 99.999% of all the bacteria in the world, all the microbes in the world. And yet, the idea of being able to understand what a neighborhood is eating in terms of ethnic foods or what have you, being able to understand that through genetic sequencing unlocks all these new ways of understanding the world. It is also worth noting that half of everything that they found doesn't even really have a name yet. This is how uninterested we have been in our own planet. And that as we set out to colonize other worlds, we actually don't even yet have names for half of all of the species of life on our own. This is the sort of kimchi story about Korean food. Anyway, so that's the Chris Mason lab doing subways. This is the Eric Alm lab at MIT. And they had the same question. Are different parts of Boston and Cambridge somehow different than others from a microbiological perspective? This was their question, what's in our collective gut? But if you thought that this was a kind of like crappy business to be in, of like swabbing the subways, it's nothing compared to this, which is actually putting robots in the sewers to core the shit mash. You know, I don't know how else to say it. To basically core the shit mash and sequence what they find there and start to understand that indeed there are unique neighborhoods from non-human perspectives. A lot of their data is sort of kept close to the, they keep their cards close for a lot of reasons, but this lens has sort of been opened up. This is Jessica Green's lab at the University of Oregon. She runs the Biology and the Built Environment Center, and so if you think about Chris Mason's lab is working at the level of the subway, and Eric Alm's lab is working at the level of the sewer, Jessica Green is working at the level of the building itself. How do we start to map the building? And this is her animation. She had a very clever idea, which is to use the filters in the HVAC systems that you can see around us right now, to basically pull those out as if they were fishing nets. They are indeed filtering out a huge number of the microbes. If you pull those out and sequence what they capture, you can start to make a map of how microbes move through a building. And if you do that, and this is sort of part of what her lab looks like, you can start to actually see what air is inside of a building, which is of course different from room to room, building to building, floor to floor. And I don't have time to get into it, but the concrete results of her work include starting to understand that we need to completely rethink, for example, how we build hospitals, which are built around the notion of sterility, of just eliminating all microbes, and so you don't get sick. And of course, all you're doing is making an environment where the one microbe that is resistant to all of that can completely wipe you out. Whereas what her work does is basically say, it is in fact statistically far healthier, rather than making a totally sterile environment, to just open the window, which I think in general in Central Europe you people knew already, but still America will get there. That's Jessica Green's lab, and then the last one was our own at the Media Lab. And I would say that although we worked with some very, very hardcore scientists, including some of the ones I just mentioned, we were really coming at this from a kind of art and cultural and design perspective. And it was inspired in part by this, which I was in my home and I would look at every day. This is a poster for the Mori Art Museum in Tokyo, the first show they ever did called Happiness by Takashi Murakami. And I would always look at this and I would, there's this sort of spiral of happiness coming up out of the Mori building, the Mori Tower. And I would always look at that and wonder, that happiness, what is it actually? And through a weird series of connections at the Media Lab, came to end up working with the firm that develops and designs and builds these buildings in particular, that had actually commissioned this poster, the Mori Art Museum, or the Mori Building Company, sorry. And it turns out that the Mori Building Company has their own ideas about a very strong integration between nature and the city. And they have done an immense amount of work around urban gardens, and this is actually, I think, the 50th floor of one of their buildings, where they've seeded a lot of trees and so on and so forth. And we sat down with them and we said, yeah, okay, yes, we agree with you that nature and the city need to be rethought, but nature is, it's both a lot bigger and a lot less visible than you think. That if you look at the entire tree of life, this was recently done about 2 years ago, if you look at the entire tree of life, the only parts where there's anything that you can even see is way, way down there. It's about this big in scale of the parts of the world of life that are visible to us. And so how do we start to think about building for the rest of that life? How do we even begin to imagine it? So, and how would we take an approach the way Attica had for Wi-Fi signals and start to bring that to this invisible kingdom? So we brought in the guys who had done this, Jack Schultz and Timo Arnall, and we spent a lot of time meeting with some pretty hardcore biologists until we met with a whole bunch of sort of like highfalutin ideas about how we could possibly do this. And then we met George Church, who said we can't do them in that way at all. That it was totally impossible to do it the ways that we needed to. We needed to actually physically sequence the material. We needed to gather genetic material and sequence it. And we had to figure out how. How do we get the material that we're going to sequence? If we don't get it by swabbing the subways, if we don't get it with robots in the sewers, if we don't get it by pulling the filters, how do we get it from the city itself? And we thought about that for a long time, and then I remembered this story in Brooklyn, which is one of my favorite stories. I don't know in Switzerland, but in Brooklyn, if you have a beard and tattoos, you get a beehive to put on top of your building, and you can get honey from it. And one of them in Williamsburg discovered when he went to get the honey that the honey was bright red, and had to figure out, well, why is it bright red? And they had to sort of like follow the bees to figure out what they'd gotten into, and discovered it's exactly what you were thinking as soon as you saw this photo, that they had gotten into the maraschino cherry factory down a little bit further into Brooklyn, and the bees had gotten into the syrup of the maraschino cherry factory, leading to an investigation around the health and human safety of that, which turned out that the maraschino cherry factory was actually just a front, a cover for the largest marijuana grow farm in New York City's history, which they had gotten away with for many, many years until the bees. And so we asked if bees could teach us something about Brooklyn, what could they teach us about Tokyo, what could they teach us about the urban environment in general, and to start to use beehives as metagenomic sensors for the environment. So we worked with beehives in Brooklyn and in Tokyo, and we worked with what's called bee debris, which is basically their trash, right? Because bees are going to travel out, they're going to gather all this stuff, and then they dump it at the bottom of the hive. So we take their trash, we purify it, we extract it, which is its own pretty intricate process, and then we take that extracted DNA and we bring it to my colleague Elizabeth Henoff, who sequences what we find there. This could be like a real-time live video, because this is like some of the dullest, lengthiest processing you could possibly imagine. You are taking one of these little vials and taking out all the DNA and looking it up against all of the possible DNA in all of the world, which is basically like handing you a bag full of shredded paper and saying, I just need to know which library this is from. All right, you know, it's really, it's some of the most ambitious computational work that humans are undertaking these days, and it's intensive, dull, but starts to unleash something. So the question was, how do we even get the material? So we set out to adapt the hive infrastructure. We built a kind of sterile tray that goes underneath the hives that was inspired by old photographic plates, same principle. So basically that goes in under the bees dump their trash at the bottom. We would yank the slide and send that off to the lab for processing. So this idea that the bees would do whatever it is that they're doing within 1.5 miles of the hive, which is what they do, and basically would give us a profile. This is what one of the hives looks like in Chicago, and so on. And we ended up doing this around the world. We did it in many sites in the different cities. We did it in Venice, which was quite illuminating about what's happening in Venice at a microbiological level, which is not great news, by the way, for Venice. In the end, it produces this massive amount of data, and this is sort of the science of it. It looks like this, and it looks like this, and it looks like this. What we were setting out to do, my particular team at MIT, was not really to publish a paper, but rather to write a poem, to figure out how to lift up the data that we were getting and use that to start to change the view of the world. So this is how we installed it at the Venice Architecture Biennale. This is the team of people, all of whom are mutants of some kind or another. This was the beehive in the window. So basically this was the metagenomic sensor for Venice, a hive full of bees. And they would go out and gather that material and they would bring it back, we would sequence it, and then we had these animations which were generated procedurally from shooting thermal imagery of the areas where we were collecting from and mapping the DNA of the things that we found. These were displayed on monitors at a dot pitch that was so tight that you actually couldn't even really see what was going on without having a physical lens on top of it. And that was in part to sort of make clear the action that we were engaged in, which is to try to open up some kind of a lens onto our own world, onto our own planet, to sort of rethink how we're shaping it. This is sort of what some of the animations looked like, and these are actually incredibly rigorously produced in terms of the specific microbes that they represent. But overall, the science that is emerging in this is giving us a huge amount of data, but it's not really giving us the imagination of this. And I think that is one of the sort of important jobs ahead for designers in the 21st century, which is, number one, how to produce the awareness of this world, and then ultimately how to design for it. This Philip Gorovitch quote, that this is what fascinates me most in existence, the peculiar necessity of imagining what is in fact real. We're not going to get there by just giving everybody microscopes and slides and so on and so forth. We need to be able to communicate this invisible scale at scale. And how we do that, I think, is up to everybody. I feel like, we started this, and I feel like, we're just beginning to see this. And overall, just to build the awareness that just like us, each one of us in the room, cities themselves have cargo that might secretly be the captain, and the question is where it takes us. So thanks very much. Sorry to go over time, and looking forward to your questions.